Thanks for the opportunity to speak to you again. This time the topic of my presentation is hybrid immunity SARS-CoV2, the new immunological normal. And I'll start by saying that I have no disclosures to make. Thank you. So as always in my previous presentations, given the fact that I'm based here in South Africa, I root my presentations within our experience in South Africa. It very much mirrors the experience of the rest of the world. So this is a slide that shows South Africa's epidemic waves and variants and kind of where we are now in South Africa. And as you will see, it'll be very similar to what you are all familiar with from many parts of the rest of the world. We had a beta wave of infections, which followed the D614G, and that was followed by Delta, and then an Omicron series of infections, which are to some extent still running, though the degree of testing here in South Africa, and as in many other parts of the world, is fairly limited at the moment. So truly we have little idea of how many infections are ongoing in South Africa and in many other parts of the world. You will all be very familiar with these kinds of schematics which show you the variants of concern which dominated each of the different waves. And in this case, they're modeled on the D614G, and each one of these pieces of red text shows you a mutation or a change that defined first beta, then delta, and then Omicron. And as you will all be extremely familiar with by now, those emerging variants of concern showed varying degrees, but sometimes very high degree of resistance to neutralizing antibodies. And that made our vaccines much less effective at preventing infection, but not at preventing, happily, not at preventing severe disease. And I'll come back to why that is the case at the end of the talk. From an immunological perspective, these various mutations really matter in how our immune systems see the variants of concern. So different mutations that are present in the variants of concern look very different to the immune system. If you're not an amino acid kind of guru, then an E484K seems like a fairly trivial mutation, for example. But an E is a negatively charged amino acid, whereas a K is a positively charged amino acid. And from the immune system's perspective, that's a big deal, enough to change the way in which your immune response sees the beta variant in this case. And the same is true when you see this kind of annotation here, delta 242 to 244, also sounds fairly trivial. But that's a 2 amino acid deletion in the N-terminal domain, and that's essentially resulted in a reshaping of the N-terminal domain. And again, that makes it look very different from the immune system's perspective, and means that the immune response has to mount a slightly different response to this variant. And of course, when you get something like Omicron, as you can see here, just focusing on the immunodominant regions, the RBD at the top and the N-terminal domain, there are lots and lots of these changes, and many of them are biologically very meaningful. So again, the Omicron variant looked very, very different compared to the other variants of concern. And partially for that reason, other reasons as well, including viral load and many other reasons, but partially for that reason, the way in which our immune systems react to each of the variants of concern is slightly different. And this is sort of a meta-analysis of work that we and others have done, asking that question: how does an infection caused by D614G or beta or delta or BA1 or BA4 change the quality of our immune responses to that variant? In this graph, anything close to the dotted line is a very cross-reactive response. Anything below the dotted line means that that infection was better at triggering responses that were strain-specific. So, for example, beta over here triggered very high-quality neutralizing antibody responses that were capable of recognizing all sorts of different variants of concern. In contrast, a delta infection actually triggered pretty strain-specific antibodies that were much less effective at neutralizing any of the variants of concern compared to their ability to neutralize delta. So without getting into too much detail, beta and BA4, for example, seem to trigger much more cross-reactive responses than BA1 and delta. And the reason we and others had done so much of this work was because we were trying to understand which of these different spikes would form the basis of a better vaccine. So different variants of concern trigger different specificities and levels of breadth, and this has implications for next generation vaccines. But really the purpose of today's talk is to make the point that these studies are no longer possible and probably no longer very relevant either. And the reason for that is that our populations, this is again South Africa, but this is a global scenario now, our populations are getting immunologically incredibly muddy. This slide here shows the percentage of people in South Africa who had been infected after the Delta wave. So that's fairly early on in the pandemic. But after the Delta wave of infection in South Africa, 73% of people had already been exposed to SARS-CoV2, either through infection or through vaccination. 73% of people had antibodies to the SARS-CoV2 spike. In South Africa, by the end of the major Omicron waves, the BA1 and BA2 waves in South Africa, 97.2% of my country had antibodies to SARS-CoV2. So virtually everybody had either been vaccinated or been infected or a combination of both. And that's very much the context in the world now. There may be pockets where it's different, places where there was an extremely hard and prolonged lockdown. There may be lower levels of seroprevalence, but this is a scenario that's becoming increasingly rare, and almost across the world, we're seeing high levels of population immunity. Of course, it had a happy outcome in the case of Omicron, and you'll all be familiar with this, where we saw for the first time a decoupling of infections, numbers of infections and numbers of hospitalizations and deaths. So despite the fact that in South Africa, Omicron was a massive wave of infections, we saw very, very few hospitalizations and deaths, as did the rest of the world, with a few exceptions. But that trend continues now. So what this means is that we have now moved into a new era where we're now tracking serological responses and T-cell responses in the context of very high levels of prior infection, and that changes our understanding. I'm going to show you a couple of examples of the kinds of work that are coming out now in that context of high levels of hybrid immunity, and try to explain what that means. And the first is a study I'm going to show you where we worked closely with Professor Cheryl Cohen here in South Africa, who had set up this incredibly impressive household transmission study with intense sampling during the first year of the pandemic. So again, this is going back a little in time. But these were two sites that were set up in Agincourt, which is a very rural area of South Africa, and Klerksdorp, which is a much more urban area of South Africa, much more densely populated. And what the first C study did was to go in literally every week and take bloods and take nasal swabs and track over a year from July 2020 through to September 2021, how many infections had occurred and how many people in that household cohort showed antibodies to SARS-CoV-2 infection. This work is published, but this is a snapshot of what happened in Agincourt, which is the rural, low-density area. And you can see during the course of this year an increase in the number of infections detected by PCR or PCR and serology. And by the end of that period, many people had antibodies to SARS-CoV-2. Even more dramatic down here in the context of Klerksdorp, which is a very heavily populated area. People are living close to one another. It's very conducive to the spread of SARS-CoV-2. And so we saw even more in this case, reported in this publication, high levels really from early on in the pandemic. And by the end of the study, almost everybody showing some level of seropositivity. So this is the kind of era, this is again work from the same consortium, but this is the kind of era that we are now in. This graph tries to use different colors to show the immune exposure that people in South Africa have had. So down here at the bottom, you can see as each of the waves come in, we saw a wave of beta, and then on top of that, a wave of D614G or wild type, then a wave of beta. But you can see that it becomes increasingly complicated over time. And by January 2022, you see a huge number of these hatched colors. And what the hatching in this context means is that people have had not just one exposure to SARS-CoV-2 through vaccination or infection, but multiple exposures, repeat exposures to the spike, either a wild type and then an Omicron infection, or a beta and then an Omicron infection, Delta, then Omicron. You can see at the end here that the Omicron wave accounts for a substantial proportion of that, but many people, almost half, have had repeat exposures. The fact that people have had this combination of infection and vaccination really changes the quality and quantity of our immune responses to SARS-CoV-2, both at the level of antibodies, but also at the level of T cells. And I'm going to show you some examples of both. This is asking the question of what happens to people who've been infected and subsequently received a vaccination. And there are many such studies. In this case, we studied a group of uninfected people in green, compared them to a group of people infected in the first wave in blue or the second wave in pink. And you can see that if you compare pre-vaccination and post-vaccination, The greens, people who have no evidence of prior infection, certainly mount an immune response to their vaccine. But in comparison, the people who've previously been infected already have antibodies, which are dramatically boosted in the context of a single vaccine. This here is a single vaccine of the adeno 26 Johnson & Johnson vaccine, and the effect that you would see would be even more dramatic in the context of RNA vaccination, and certainly even more dramatic in the context of multiple RNA vaccinations. And this is now well documented in the literature. What about the other way around? Although we have now, everybody has some level of seropositivity, you've got to remember that almost everybody has fairly low levels of antibodies. In SARS-CoV2 infections and COVID-19, the more severely ill somebody is, the higher the antibody responses are. But as you all know, most infections are either asymptomatic or mild. As a consequence of that, many people experience a breakthrough infection, so they have some sort of antibody responses and nonetheless become infected. And that too results in dramatic increases in antibody responses. This is a work that we did actually, looking at a cohort of healthcare workers who were vaccinated with the Johnson & Johnson vaccine and then subsequently suffered a breakthrough infection. And the impact of that breakthrough infection on antibody results is shown down here. In blue are people who received the vaccine but never suffered a breakthrough infection. You can see those are fairly stable and low. But over the pink time frame, you can see individuals who did suffer a breakthrough infection, and you can see an incredible increase in their neutralizing antibody titers. Transient, and they start waning, and we can come back to that. But you do see this incredible increase in neutralizing antibodies. So if you think back to the graph I showed you with all the different colors, almost everybody falls into the category of either having had a breakthrough infection or having had multiple exposures on top of one another. And almost everybody now is likely in this group. It's not just a case that hybrid immunity increases the quantity of your responses, what we refer to as potency, but it also increases the quality of your response, what I would describe as cross-reactivity or neutralization breadth. Here, this graph compares people, again in blue, no breakthrough infection, with those in red who had the breakthrough infection, and asks how good their antibodies are at recognizing both the virus that infected them, but also lots of other variants, including right at the end here, SARS-CoV1, which clearly these people have never seen in their lives. And you can see that hybrid immunity in this context, a breakthrough infection or a BTI, means that these individuals have such high antibody responses that they are even able to neutralize SARS-CoV1. So they are not only potent, they are also cross-reactive, and that is likely a consequence of repeated exposure, which means that people's immune systems have had time for the antibodies to undergo affinity maturation, somatic hypermutation, and become much better at neutralizing SARS-CoV2. Does that mean that we are going to see the end of breakthrough infections? I'm afraid not. This is a breakdown from today, looking at the prevalence of different SARS-CoV2 sequences. And you can see that the most dominant variants at the moment are BQ1 and XBB.1.5. And we know that these variants are extremely neutralization resistant, and that even high levels of antibodies, which is the norm now for our populations, may not prevent all infections. Will certainly prevent some infections, but not all infections. What about the T cells in the context of hybrid immunity? Initially in the pandemic, we focused very much on neutralizing antibodies, but it's the T cells that prevent severe disease and often prevent deaths in the context of SARS-CoV2 infection. Unlike neutralizing antibodies, which are really severely impacted by those mutations that I described in the first two slides, the T cells are really resilient. Part of that is that they recognize many more aspects of the SARS-CoV2 spike, not just those immunodominant RBD and NTD regions. But for many reasons, these T cells are able to retain their activity against the variants of concern. And that's shown here for T cell CD8 responses against Omicron. Down in this graph here, anything which falls down dramatically would be a loss of activity, but you can see that somewhere around 80% of the CD8 response is preserved. This is work from my colleagues, Catherine Rue and Wendy Burgess. So these killer immune cells still recognize the Omicron variant. What are their levels in the context of these multiple exposures that we've been talking about? Well, it's slightly different for the T cells. I'm using one publication here, comparing both antibodies, which I'll show you shortly, and T cells in the context either of 1, 2, 3, or 4 exposures. either vaccination and or infection, a real hodgepodge here of exposures. But if you look at the antibodies, as I've shown you in the previous slides, those antibody titers go increasing, as you can see, from fairly low levels early on with one exposure to really maxed out at the top here in individuals who've had 4 exposures. The T cells are much more stable in responsiveness to multiple exposures. You do get many individuals after a single exposure who have CD4 T cells, shown in the blue here. Fewer people who have CD4 and CD8 T cells. CD8 T cells are less common in the context of SARS-CoV2 infection, that's well known. But you can see if you focus on the individuals who had CD4 and CD8 T cells, it doesn't increase very dramatically despite the number of exposures. So unlike antibodies, which get better and better, just certainly during exposures to the virus, the T cells are much more stable, but much more durable against the variants of concern. So this is where we are really, is that hybrid immunity is our new normal, with most people having been exposed to the spike protein and or the entire virus, which impacts T cell responses particularly, many times through a combination of infection and vaccination. Population antibody levels are sky high at the moment and broad, with multiple exposures having driven affinity maturation. But what we don't really yet know is durability, particularly as population spread changes. And it may not matter very much because it's the memory responses we really care about. We do know that breakthrough infections will continue, and emerging variants of concern show exceptional resistance to neutralization. And T cell responses are very common now at the population level. And while they are more stable, they are less impacted by variants of concern and will likely continue to protect from severe infection. With that, I'd like to stop and thank my wonderful lab and our funders. Thank you.